I was saying that I'll just go over briefly the study guide, and then questions that I feel like, or synthesis questions that I think that you want to focus on for each lecture or each set of study guides. Yeah.
Okay.
And then if you have questions, you can just ask me throughout, and I can specify more about those type of questions.
Okay. Is the percentage for like the previous lectures, like 1 through 11, is it half, like 50% would be on the final? I thought it was like 25%.
So lecture 1 through 11 is going to be 50%, right? Because the other half is cumulative. How they're going to be split is going to be between the first and second exam, right? So first exam, you had lectures 1 through 6, right? So you'll probably have about close to 25% of the questions coming from that. And then the second half, which is lectures 7 through 11, right, is where you're going to have about another half.
Okay.
Right. So combine the first half of the class, lectures 1 through 11, is going to be about 50%, and then the rest is about 50%. Okay. So it's going to be, again, just because for clarity, just a reminder, I expect everyone to show up for this anyway, but it's going to be Wednesday, 8 a.m. to 11 a.m. People who have extra time will have that extra time. And it's going to be 3 parts. I am pretty confident that all of these sections can be completed within that 3 hours. And the format is going to be the same as previous exams. So you're going to have multiple choice, part B and part C. And I'll throw some extra credit questions in there as well. Again, it's extra credit. It's really up to you whether or not you want to go back and answer them, depending on your time. So part B, again, it's going to be short answer questions, right? Fill in the blanks, true, false type of questions. Primarily, that's what it's going to be. And then, or just a single answer question, single answer. While part C is going to be it's a little bit more answers. So it'll be a couple of sentences or kind of a more, I guess, synthesis of the material. Madison, you have a question?
Yeah, I just have a quick question. I noticed that our study guide for lectures 1 through 11 is like very short. Do you just recommend that we focus on these topics first and then brush over everything else from lectures 1 through 11? Yeah. I'm just a little worried because there's so much information, through 1 through 11. I don't want to leave anything out.
Mm-hmm. Mm-hmm. Yeah, so I'll get to that in a minute, but yeah, definitely I would, that was the intention of me compressing this lecture 1 through 11 was for you guys to really focus on those concepts, because that's where the majority, if not all, of the questions are going to be coming from.
Okay, thank you.
So that's as we're going to, the next question, the next slide relates to that. So this is really based on what I expect to be on that exam. That's from lecture 1 through 11. So these are the concepts, right, that I really will test you guys on, or you will see again. So you have to know how to design a primer. That's a given, right? And that can be, when I say know how to design a primer, that can be could be like a multiple choice question or a question where you're given a sequence and asked to design a primer. And that's really the extent of it, okay? Paying attention to the fact that you always have to do the 5' to 3' orientation. And if it's a case where you need to have a forward and reverse primer, then you need to have a forward and reverse primer for these. The basic unit of chromatin and what is chromatin, right? You've seen this in lecture, in exam, in the second exam, I think, right? So you've seen this in the basic unit of chromatin, right? You know you need your histone proteins wrapped around your DNA, right? That's kind of really the extent to understanding chromatin that I want you guys to understand, because this is a very general concept, right? What is chromatin, right? This is really where your DNA is compacted, and it's compacted because it's wrapped around these histone proteins in the cell. chromatin remodeling complex. What exactly you need to know for chromatin? What are chromatin remodeling complexes? What they do? What do they do? We had a question in exam 2 where, and many of you had the same challenge, right? When I asked the ways in which you can alter the chromatin structure. And many of you thought, okay, you have all 4 of these possibilities where the chromatin remodeling complex will change the chromatin in a way, in some way, either by sliding or replacing the histone proteins, etc. That is really one way, right? Is using that chromatin remodeling complex to change the chromatin structure. And the other way would be that you need to modify those histone tails, right? Because that's really what you're doing. By modifying those histone tails, you're altering the chromatin structure. So really understanding the difference between those 2 things. So histone tails modification, whether it's your acetylation or your methylation, right? That's one way. And then using these chromatin remodeling complexes is the other way that you can alter the chromatin structure. That question actually didn't even require that you list nucleosome displacement or nucleosome sliding. That wasn't even required. I think what was required was saying just using chromatin remodeling complexes because it's understood what that means. So try to think about it in terms of the bigger concepts, right? For the questions that I need details for, I will specify details where I expect the details to be known, but otherwise, if it's a major concept, then you have to just think about it more on a larger scale. You'll see the LAC operon and TRIP operon again integrated in multiple choice questions or short answer questions. You're not going to see this in part C, right? So again, understanding how the LAC operon and TRIP operon is regulated, meaning what is the role of an inducer, for example, right? What happens if you have lower high levels of lactose and tryptophan? That's, again, that's the extent of what you need to know. So you're not going to have like a diagram where you have to go through and do those examples again. Components of PCR reaction. Again, there's certain things that I really want you guys to get out of this course. I mean, so you need to know what the component, and one of them is PCR, right? What are the components of a PCR reaction? And I think for the most part in the previous exams, most of you were, right on mark with this. I mean, because you obviously knew this. But for people who didn't get all of those components correct, you want to go back through and know this, yeah, because you will absolutely see this again on the final exam. Composition of telomerase, another question. So this is something you will see again in the same kind of context, basically asking you what is the composition of telomerase, right, and knowing what that is. So spliceosomal complexes, and the reason that I like to ask about complex, what's in the complex, is because it's important in how they actually function, right? So whether it's in a spliceosomal complex, right, you have your spliceosomal RNA and also the proteins, right? They do something, right? So knowing what those compositions are helps you to understand what are they actually doing, right? What is the role of the spliceosomal RNA, right? You know that they bind to certain pieces of the RNA to help with splicing, right? Depending on which ones you're looking at. And knowing the events that occurs during splicing, right? Where another question that you saw in the last exam, where you had splicing occurring and then the joining of the exons. Just understanding what's happening there is super important. Knowing that, okay, there's that stage where once you splice it out at the 5' splice site, you form this lariat structure, right? And that lariat structure is important. You need a branch point, which is important for that to occur. And that's an important step in splicing as well. Yeah. The process of DNA replication in the molecules, this is not in the first exam. What we did was we gave you guys a graph. There were questions distributed, of course, related to DNA replication and the polymerases. But what we did in the larger part was where we had this graph, right? And you had to identify what are these structures on that image. This is not going to be the same, right? I mean, of course, there's just not enough time to ask those questions. So it's going to be more about what are, what is the enzyme that's involved in unwinding the DNA, or what is the enzyme used if once they're to relieve the tension in the DNA, right? So it will be more in a question format that you will see. Okay, so just knowing what those parts are, I would recommend. You will not see again the difference between the different polymerases, so you don't need to worry about that. What are the three basic components of a vector and their function? To be very specific here, you need to know what the components of the vectors are, and the three basic components of the vector and the function. So in general, in a vector that's used for cloning, people often assume that you need to have this screening marker, right, which is your beta-galactosidase. We went over a lot of lectures talking about LAC-Z and blue-white screening, but that was an example of that type of vector. I mentioned that not all vectors have a screening marker. Many of the vectors we use currently in our lab don't have a screening marker, but they need to have three other important things in order for them to work, right? So you need to make sure you have your origin of replication. You need to make sure that you have a place to clone your gene in that vector, and that's the cloning, the multi-cloning site. And you also need to have a selectable marker to be sure that you've successfully transformed your plasmid into bacteria, okay? So really knowing what those three things are and how they function is important, okay? How to read a Sanger sequencing map and the components of a Sanger sequencing reaction. The components of a Sanger sequencing reaction is going to be very similar to a PCR reaction, except that you're going to have one additional component. And I'm not going to tell you guys what that is because I assume that you all know what that is. So knowing what the components of that Sanger sequencing reaction is, and how to read a sequencing map. You will see that again, right? Because again, these are kind of really core fundamental aspects of molecular biology. So you will be given a map and you will be asked to write the sequence 5' to 3' of what the sequence is, and also of the template strand, or either or. I haven't decided actually whether it's going to be one or both of them. So, but you just need to know how to read that map and how to write that sequence in the correct way. You need to know how to draw a restriction map, right? Very similar question to the question that very similar to the question you saw in exam 1, right? Was this exam 1 or exam 2? I can't remember which one it was in. This was a while ago.
It was exam 1.
It was exam 1, right? Yeah, I think it was exam 1. I just feel like I just graded those, or went over those exams. That's why I thought it was exam 2. But understanding how to read a restriction map or how to draw one, I think that is something that you should know. And I'm just telling you this, in the past I've done this, I just haven't decided if I'm going to do it yet. So, but knowing how to do it, I don't want to tell you not to think about it, because I don't know yet if I'm going to add it in there, but I've done it in the past on final exams. So I would suggest that you just know how to draw a restriction map based on similar to what you saw in the last exam or what you've done in your discussion classes. From all the way from lecture 1 through 11, and also some of what we saw later on, right? We talked about different methods to look at protein and nucleic acid interactions. I added nucleic acid because we did a lot of protein DNA, but it can also be the same for protein RNA as well. But looking at how some of what those type of interactions are, and if you were given a question on, how do you determine whether or not your protein binds to this particular DNA sequence that you just discovered, what method would you use to do that? Okay. So that's for lecture 1 through 11. Any questions about that? And really, I would focus on these particular things that are listed here. And if you feel like you want to go back and you have time, fine, but I would highly recommend you just focus on what's listed here. I mean, it's 50% for both lectures. There's only so many questions that I can possibly ask. So again, you'll see that a lot of the exam is really on the last set of lectures. It should be 50%, give or take, but it's also going to be kind of integrated. So I would really just focus on these particular concepts for lecture 1 through 11, because this is what you're going to see again. And some of the extra credit questions are going to be exactly the questions that most people had challenges with on the previous exam. Yeah, and those are going to be the questions that are extra credit. Or I might take them from the extra credit or poll questions. I haven't decided yet, but there's going to be questions that you've obviously seen before in some format. Okay. So that is for lecture 1 through 11. Now, for the what we haven't been tested on yet, so lecture 12, I think I would follow this study guide. Again, a lot of the questions are going to be coming straight out of these study guides, the concepts that I've listed here in the study guide. And I really hope that you're able to go over this material. Let me know if you have any specific questions by sending either Israel or I an email. You need to understand what is RNAi, possible outcomes of regulation, meaning if it's RNAi, what can happen at the transcription versus post-transcription versus translation level, right? Depending on the organism, you can have different outcomes at the mRNA or the translation level. Knowing the types of and the roles of the non-coding RNAs, small non-coding RNAs in gene regulation, right? We talk about small RNAs, microRNAs, and these piRNAs, meaning what do they do, right? I mean, most of them, there was in those slides, they list what the function is, right? I mean, so just knowing in general what the function is. I'm not going to ask you to draw a pathway, or I'm not going to ask you to draw anything for these. I mean, so just understanding at a basic level what they are, how do they regulate gene expression, what are some of the proteins and nucleases involved. And for these, we talk about the ones that we talk about the most that are highlighted are the ones you need to know, like what is dicer, what does dicer do, right? What is Argonaut, what does Argonaut do, and what is the RISC complex and what do they do? And because I like to ask about complexes, you want to also know what the components of the RISC complex is, right? And you'll know that that also includes Argonaut as well. So that's really, I mean, for RNAi, I mean, there's only so many questions I can ask about that. Like, there was a lot of details there, but please try to focus on how is it triggered, right? Double-stranded RNA, double-stranded DNA, how does it get triggered? And then what happens once the cell encounters these double-stranded RNA molecules, okay? And in the sense of the CRISPR technology, here again, very similar to RNAi. What is the technology? What is the difference between CRISPR and RNAi, right? So how does it get triggered in the cell, right? Using these two different pathways, and what's the outcome, right? Like, what's the difference between CRISPR and RNAi in terms of what is it doing in bacteria versus other eukaryotes? And then also the way in which we're now using CRISPR to alter genomes, right? How is it being used? And to be able to do this, I want you to think about how you may use CRISPR to edit a gene that you might be interested in. That relates to this slide, right? So if I asked you list 1 or 2 ways in which you can use CRISPR to modify genomes, this is what I'm referring to. So I suggest you go over this slide and think about what is happening in this pathway, primarily what's happening in A, right? Because this is the very classic way in how we use CRISPR to alter the genome. Understanding that you need to, what is the composition of that CRISPR complex, right? You need your Cas9, you need your guide RNA. Together, the two of them get to the DNA. It's important that you recognize this photospacer adjacent motif, this PAM site, which we mentioned previously, is any nucleotide plus 2 Gs after that, right? So NGG. These are absolutely critical. So if I ask you again, what are the features of to design a CRISPR experiment, I expect you to tell me Cas9 guide RNA, and you're having this PAM site within the target DNA where you're going. Okay, when this happens, Cas9 will cleave, create a double strand break, and because the double strand break is going to be repaired, most of the times it's going to be repaired by non-homologous end joining. And you remember when we talked about non-homologous end joining, because of the way it works, you have to remove some bases, right? And that can create, by deleting bases, you can also accidentally insert bases. You can create a mutation because you're adding DNA that wasn't there before. And if this is in a gene, that will alter the gene because it's going to cause a frame shift, right? Some kind of a frame shift mutation that we talked about before. Yeah. So understanding how to do this. So the type of question that you can expect to see, and this is from previous exams as well, is where you will be asked, you will be given a question or a scenario, right? So you have a protein sequence, right? And you will be asked to design or write the guide RNA primer, right? Because people often get confused. Yes, it's going to be a guide RNA primer because it will be converted to RNA later on. But when you design the primer you're designing is a DNA sequence, right? So write the DNA sequence in the 5' to the 3' orientation of that guide RNA to bind to the region within this protein, okay? I will give a hint because, and I usually don't do this as well, but I'll give a hint that you want to start with that PAM sequence. So you need to look at your sequence and find 3 bases where you have some nucleotide, which N can be anything, and 2 Gs, okay? So in this context, if you had to design a primer in a sequence that's like this, right? So you have your sequence, you scan your sequence, right? And you're looking for, I would say look for the 2 Gs, right? If you scan this entire sequence, here you have 2 Gs. I told you it has to be NGG, right? So this is where you will be start designing your primer, right? This is 3', this is 5', right? So your primer needs to be 5' to 3'. So understanding how to do this. This is, if you know to design primers, this is going to be easy to do. So it shouldn't be that challenging, but this is kind of what I will ask, the type of experiments you can expect, or have a question related to experiments that you can expect for these CRISPR-Cas9 type of experiments. Any questions about that before I go on to the next lecture? The next lecture, study guide for the next lecture.
Yeah, so when you say like design an experiment for the CRISPR, is it just kind of like explaining it in detail? Or like, or would you just give us like a scenario, kind of like this
I give you a
question here?
scenario. So I'll give you a scenario and I'll ask you first to answer specific things, right? So here you're given a scenario and I'm asking you to design the primer for the guide RNA.
Okay, that makes sense. Thank you.
Mhm. And so for lecture 13, so the answer will, all the question will, someone asked if the question is going to be in the form of DNA. So you will write it in DNA unless I specify to write it in RNA. Okay, so I'll be very specific in the question. So you just write what I asked, yeah. So what are, for lecture 13, what do you need to know for lecture 13? Oops, sorry. Okay, so for lecture 13, you have we talked about translation, right? The translation machinery in both eukaryotes and prokaryotes, how similar and how different they are, right? And really, it's really the number of the initiation factors and the ribosomal subunits, right? They're different sizes, but the concepts are pretty similar between these two pathways. Also, the way that translation gets initiated is going to be different, right? And we'll talk about that in a little bit. So part of understanding translation initiation is really understanding what happened, what needs to happen first, right? So if I ask you, okay, before the ribosome can attach to the mRNA, and the tRNA can also find the start codon, what needs to happen, right? And we talked about those two important events that needs to happen. So understanding what those are, right, which is pretty much here. So that you need to have that the tRNA charging step, right? And what this charging, what does that mean? What are you doing? And I don't expect you to know every single step, meaning I don't expect you to say, okay, you're adding ATP and this is what's happening. Just a basic concept of what is tRNA charging? What are you doing with the tRNA? And how are you modifying that? And particularly where that modification happens, okay? And then once you understand that, understanding what happens to the ribosome, right? Because the ribosomes are usually together, so you need to understand that they get separated and how are they prevented from getting back together, right? So kind of thinking about still along those steps. Denise?
Yeah, so just like the elongation, for translation, the process, do we have to know like in detail what's going on? Like, oh, ATP is hydrolyzed, or if we just, because I think there was like a specific slide and it kind of like shortened it, but then like the following slides was like more in detail. So I don't know.
Mm-hmm. Yeah, so for translation elongation, you would need to know that, I would say focus on the steps where it's shortened. We went into more details for the details of that step, but I don't remember, I'm sorry, I don't remember exactly which slides you're talking about, but I know the ones where it's shortened, where it was like 3 major steps that you need to focus on. And I don't remember what slide was after that, but it, if there is a particular factor that I want you guys to know, and I'll probably tell you what that is in a minute, then I would focus on that. But I think understanding the 3 major things that's happening in transcription elongation is what's important. And I'll get to that in the next slide, actually. So
yeah, I thought this was 13. Okay.
Yeah. So I mean, the next slide, meaning I have a graphic which we can talk about in a minute. So so and then you can let me know if you have other questions about that. So again, understanding how you build this 30S and 40S translation initiation complex, right? What are the components of building that complex to start? How does the ribosome know where to start translation, right? It's going to be different between prokaryotes and eukaryotes, and knowing what that difference is. I don't expect you to know the consensus sequence for the Shine-Delgarno sequence, because that can, that's going to vary. In terms of the Kozak rules, I'm trying to remember besides knowing that what the Kozak rules are and how they're important, do you need to remember what's happening? I don't think you need to, I don't think you need to remember exactly what's happening with the Kozak rules, like where you have a +1 position and then whatever else is downstream of that at +3 position or upstream of that. But knowing what the Kozak rules are, I think that's definitely is going to be important. Yeah. Okay. And how does the ribosome bind to the mRNA? Yeah, what interactions are important? Again, this is going to be different between prokaryotes and eukaryotes as well, right? So knowing what that difference is. What are some of the consequences of mRNA secondary structure? So that same slide where we had what happens if you have a hairpin structure in a certain position, how does that affect translation? And for all of these components, this probably gets back to your question, Denise, is, there's lots of proteins that are involved in different steps for the elongation part. For the initiation and getting everything assembled, for bacteria, it's much easier. So it's 1, 2, and 3 initiation factors, which are very straightforward. With eukaryotes, getting all of that stuff assembled when you're making this 40X translation initiation complex, but then this gets converted in the end to like 48S, right, before you begin. There's like 5, I think, different elongation factors that play a role. But there's some that are super important, right? More than others, right? The one that keeps the ribosome separated, the one that helps to recruit the tRNA with the mRNA to that ribosome, right? And also the one that helps to scan the ribosome to find that 5' cap. So these are kind of the ones that are super important. Because I think in that step, they also have like EIF1 and 1A or whatever. I probably wouldn't ask anything about that because I shouldn't say probably. I know I'm not going to ask anything about that because they're not as relevant to the overall process as they would like you guys to know. Knowing what EIF4 is, and I said all of the subunits of this complex. So in elongation, right? And because I'm jumping ahead of myself. So in elongation, you have all of these different things that are happening, right? In order for the ribosome to scan along and find that 5' cap, you need to have this EIF4F, which is that cap binding protein, right? And within that cap binding protein, you had, I think it was EIF4E, A, and G, right? And that's the subunit complex. I'm not going to ask you what each one of those do, but I definitely will ask you about EIF4G because that's a scaffold protein that's super important for many other processes later on. Yeah. So knowing what EIF4F does and what EIF4G does, I think would be important for you to know. And then in terms of within that steps of elongation, I told you this slide had like 3 different steps. Those are really the steps that you need to focus on in what's happening, right? So you're bringing in your new amino acid with the help of this EF2, right? That's important because that's what brings it in. Peptidyl transferase is important for attaching these 2 together. And then you have translocation that's due to this EFG that helps to translocate to the next position, right? So there's really 3 things that are happening. The new amino acid coming in with the help of this protein, it attaches to the amino acid that's there, right, to grow the polypeptide chain, and then it gets translocated and that step gets repeated. So it's really 3 main steps that's really happening. The type of question or a type of question that you can see incorporated into the fill in the blank types of question or a more longer question. Again, in both prokaryotes and eukaryotes, thinking about how many tRNAs are usually active in translation at a particular moment, right? So, and this is mostly during elongation that this is happening to some extent, right? Because once initiation starts, it's just, you just have your 1 amino acid that's present there. And then as translation is proceeding, meaning it's active, so you have elongation happening, you really think about how many amino acids, how many tRNAs will be active at that site. So just a wild guess, right? I mean, and it's there, it's kind of obvious, right? You're gonna always have 2 amino acids that are present, right? Because it's gonna get added, this shifts over, right? This tRNA goes away, and then the other one comes in, okay? So this all will not, all of this will definitely not even get started if it wasn't for EF2 or EFTU, right? Because this is the one that's needed to bind that amino acyl tRNA to that A site, right? So if you mutate this or you get rid of this, then you're really stalling translation, right? Because it's just going to be stuck. There's no other amino acid can come in. So another type of multiple choice question based on what I just said could be, during translation elongation, you accidentally deleted the EFTU protein, and what could be the possible outcome, and you'll be given different choices, and you will know, again, based on that, which of those choices are correct, right? It's not going to be because, for example, the peptidyl transferase cannot add the protein, right? That's not going to be a reason, right? Because you're stalling everything up in this step, so you want to think about that. So that's for lecture 13. Lecture 14 is very straightforward as well. We continue talking about translation, elongation, and termination here in this lecture. We also talked about ways in which you can regulate translation, right? We talked in bacteria, you can regulate translation using these heat shock proteins. And in eukaryotes, it's the phosphorylation of EIF2. I'm just thinking what type of questions I would ask about that. I mean, I think for something like in terms of ways in which you can regulate translation, it would literally probably be question, list 2 ways in which you can regulate translation efficiency in eukaryotes, right? And one of them would be phosphorylation, and the other one would be heat shock protein. But those are 2 ways. Each way is specific to a particular organism. So that I don't think I asked too many questions about this particular step, besides knowing that you know that you can do this, and if you had to do it, this is the way that it would be done. Understanding the genetic code, right? And the 2 key features of the code, they're non-overlapping, there's no commas in the mRNA. You will be given a DNA sequence. You will be asked to write the RNA sequence. You will be asked to write the sequence if the code was overlapping and if the code was non-overlapping, right? You will be given this. So knowing what that means. I don't think I'm going to ask you to give an example of a no commas in the mRNA, because obviously, I mean, that's not really that's not a realistic scenario. Knowing the regions of the tRNA that's important in translation elongation. You guys just had elongation and growing of the polypeptide chain. You kind of had an extra credit question related to this, right? Knowing the parts of the tRNA that's important in translation elongation. We talked about 2 parts of that structure that's really important, and the 2 others we didn't talk about. I already mentioned the translation elongation process as it relates to the previous lecture. We talked a little bit what's happening at the EP and A sites, right? Where does the first amino acid sits in these sites, and where does the incoming amino acid come into? Peptide bond formation, how does that happen with in terms of knowing that this is done with the peptidyl transferase, and also how that transfer happens, right? And this is stemmed from a question asked by someone during class, right? Asking if there's a common way in which those peptides get added, right? And you remember that the peptide, the amino acid that's in the P site always gets added to the one that's in the A site, and then that shifts over, right? So knowing that that's how the process works is important. Translation termination, what triggers translation termination, right? Knowing that these are stop codons, what the stop codons are. If there's any time that you need to write out the actual codon for these stop codons, you will be given that table, so you will know it. But knowing what happens when the ribosome encounters a stop codon, right? The stop codon doesn't code for amino acids, so then these release factors come in and sit there and then triggers the process to destabilize that complex and release the polypeptide chain. Okay. I also don't think that I usually ask people to know the difference between the release factors and which stop codon they recognize, because it's different with prokaryotes versus eukaryotes, right? I mean, I don't think that's something you need to worry too much about. I think knowing that when the ribosome encounters stop codon, these release factors come in and sits in that A site because there's no amino acid that can be there, and helps to destabilize the complex and release the polypeptide chain. What are frame shift mutations? In terms of what are frame shift mutation, understanding that this can occur from any of those multiple steps that I told you on that slide. So either due to an insertion or a deletion. So that was a slide where you had like all the cat codons or whatever. So if you're given a sequence and I say, okay, you inserted a base at this position, how does that get shifted into what are the next codons after that, right? So that's really the idea behind a frame shift mutation. And that can be either an insertion or a deletion. I'm not going to give you anything complicated by crossing two different lines or anything like that, right? It will really be a situation where you're inserting a single base or deleting a single base, or what happens if you delete 3 bases, right? Or insert 3 bases. You can think of that as that's not really going to cause a frame shift mutation. It's just the first codon that will be affected. But then after that, everything else should kind of remain the same. Right? So you'll have this extra codon in the middle. So strike this out with a stop codon suppression. I think I'm not going to ask about this simply because I felt like I didn't go over this in as much detail as I would like. So I don't think I will ask any question. I shouldn't say I don't think. If I say I'm not going to ask, I'm not going to ask. So I would not focus on this and try to understand this. Just knowing what is a frame shift mutation, I think, is what I would like you guys to know. Denise?
Yeah, it's just because there was a slide that kind of confused me about the overlapping in mRNAs. I mean, no commas. Can you just explain what the no commas was? I know this was a more kind of like straightforward thing, but.
So I don't have the slide, but I'll tell you what I remember. So if there was a comma, like if you have your codons, and if there was a comma after the first 3, right, then you wouldn't change anything. So you wouldn't really change the codon. So what will be happening is that the tRNA is going to read, oh, this is a codon. And then there's a comma. And so there's nothing there, right? So it will probably just skip over that because there's nothing there, right? So because it's a comma, right? But it's going to stall. So you'll know that if you were to have a frame shift mutation, and maybe I can draw it and hope I can explain this better. So let's say you had, I don't know, let's say you had a codon, like something like this. Right. So we talk about, okay, non-overlapping versus overlapping, right? Where you're going to have 4 options. If there was a comma, let's say there was a comma here, and you were to read this codon, right? So this is going to be a codon, this is going to be a codon. If there was a comma here, then if you were to have and this is directly related to mutations, right? If you had a comma here, and let's say you had an insertion, something that happens there, what happens is now you have this insertion, you have your codon here, and then you would have this weird two bases that's kind of a codon because this comma will be counted as, I don't want to say as a base, right? And you know this would be weird, but this is not what we see based on what we know when we look at the sequence in the DNA. So that's why it said there's no comma. So that's a situation where that statement is based on the fact that we were checking to see what is that composition of codons in your RNA, right? So we checked and we knew that, okay, if it's either, if there was overlapping, this is what it would be. If it's non-overlapping, this is what it would be. And we know that it's, non-overlapping because when you look at the RNA and then you look at the protein that you get, you know that that cannot be the case, right? So that's kind of why that statement says, okay, we know that this is what a genetic code is. Yeah. Same thing is experiments were done with the assumption that could there be a comma between these two bases? So if you create a line theoretically and assume that there's a comma, when you look at what happens with this mRNA, when that protein gets translated, you know that it cannot have a comma because otherwise without a comma, you would have a very different outcome in terms of the amino acids that represents this RNA. So those two statements are based on experiments that were done to determine what is the genetic code, how does it look?
So with the frameshift mutation, does it just affect like the first codon? Because I know in the slide, like it said, like the rest of it.
It depends on the mutation. It depends on whether it's an insertion, deletion, and where it is.
Oh yeah, right.
Right?
Okay.
So it depends on where it is. And it also depends on if it's 3. So like if you had, in the case, if you had a 3 base pair insertion or deletion, then the codons downstream of that are going to be the same codons, but you're going to have at that site something different that's happening. Yeah.
Okay, thank you.
And after this, I can pull up that slide again just to help to clarify if you still have questions about that. Let's move this. So that's kind of what I want you guys to know for this one. Again, type of question that you want to think about in terms of pulling all of these EIFs before. The reason I put this here is because someone asked in a previous office hour, there's so many EIFs, do we need to know all of them, right? And as I mentioned, you want to know the EIF for F because that's important. You want to know EIF for G. So that's also another one that's super important, right? So the one that helps to keep the ribosomal subunit separated, right? I mean, it's good to also know that one. So this is kind of the question you would ask, and this is, the eukaryotic initiation factor, EIF, whatever, is a cap binding protein that allows a particular ribosomal subunit to bind to the 5' of mRNA, right? And if you know that step in that assembly translation initiation, you would be able to easily answer this question. Yes, Denise. You still have a question, Denise, or your hands?
Oh, sorry, I forgot to lower it.
Okay.
Thank you.
Right. So in this case, you guys know which of the EIF 4s we're talking about, right? Because I just told you, so you should know which one I'm talking about here. Right. So which of the EIF 4 is a cap binding protein? You'll notice by, and I know you'll notice by Wednesday, right? So yeah.
Would it be the EIF4F? Like,
yes, exactly. And which of the ribosomal, so what does it allow, which ribosomal subunit to bind to the 5' end of the mRNA?
it should be the 40th one, right?
Yes.
Okay. Do you want us to be specific? Because I know like in the steps it was like oh, when EIF4F it converts 43 to 48 or something like that.
No, I
Okay.
mean, you have to be, it's really, this is really specific. There could only be two answers here, right? This can
Yeah.
only be EIF4F and this can only be 40S. Yeah.
Okay.
For lecture 15, which is more post-translational modifications. I mean, there was also a lot of details, a lot of things where I feel like you just need to know it. Yeah, what are the steps to create a functional protein, right? So you know that the protein needs to be folded properly, right? If you just say, okay, your protein needs to be folded properly and it needs to be targeted to the right location, right? Those are really the steps to create the functional protein. What is the function of molecular chaperones, right? These are the proteins that help the proteins to fold. There's a difference between HSP 60 and 70. Know what the difference is because you will be asked what is the difference between the two, okay? And really the difference between the two, don't get bogged down by those graphs showing you the barrel and the protein going into the barrel and with the help of ATP, it's being folded or anything like that. Knowing when HSP 60 is active and when HSP 70 is active is sufficient. Knowing what is the proteasome, right? And how does it work, right? So this is a stepwise process, right? Where proteins that have gone through the folding process has tried to refold it, and then in the end, you just cannot fold it, and you just need to get rid of it, right? So, and that's what the job of the proteasome is to do, is to degrade proteins that are unable to fold. And it does so by chewing them up. But in order for them to recognize those proteins that the cell have tried many times to fold and just have not been able to fold, compared to proteins that were just produced and haven't even gone through the folding process, you need to have that ubiquitin chain, the polyubiquitin chain attached to these proteins, right? Because that's what it tells. That's what the proteasome recognizes. These tags on these proteins that tells that these are the proteins that needs to be digested or chewed up or degraded or whatever. So knowing how nonsense mediated decay works, or on what triggers it, right? I mean, this is also related to the mRNA, right? So if the mRNA is not perfect, after it's finished processing, it's going out into the cytoplasm. So during that nuclear export, right, it gets scanned by these ribosomal proteins, and you go through this first round of pioneer translation to check to make sure everything is fine, to check to make sure that it's been spliced out, the caps are there and everything. So if for some reason those ribosome is scanning that messenger RNA and realize that the proteins, the exon junction proteins, are in the wrong place, right? So that kind of tells the cell that something is wrong. And the reason it will be in the wrong place is because for some reason you have a premature termination codon before that S-RNA intron junction. I'll post this recording after I'm done, yeah. Okay. So knowing that, and then protein targeting, very basic. How does it work? How does it know where to go? And that's because there's signals in that mRNA that tells you, the signal in your DNA that tells you that your protein should be nuclear localized. It gets transcribed into mRNA, and then when the protein is made, that the peptides tell it that, okay, you need to go to the nucleus, or you need to go to the ER, or you need to go, to the mitochondria or somewhere in the cell. Okay, so just knowing basic that they are, if I ask you, okay, what is present on what needs to be present on a protein for that protein to be able to be transferred to the nucleus after it's made, and you will tell me that, oh, because it have a nuclear localization signal, right? So that's what you will tell me. So you might not tell me this now, but you'll tell me this by Wednesday when you have that exam, okay? Okay. So synthesizing all of those questions, what you can expect to see for sure, and this is in the longer answer questions, and it's not going to be exactly like this, but it's a version of this that I want you guys to think about. So you will be given the table, because I don't expect you guys to memorize this, so please don't spend your time memorizing these codons. You'll be given a table. You will be given the double-stranded molecule. Okay, let me take that back. You'll be given a table. You will be given probably a DNA sequence or parts of a DNA sequence, okay? From these parts of the DNA sequence, you would be expected to know which is the coding strand and which is the template strand, okay? These are two strands. One, when I mean what is the coding strand and what is the template strand, it directly relates to the type of mRNA or what the sequence of the mRNA is going to be, okay? So depending on which of these are the coding or template strand, then you will have an mRNA sequence. That should be also 5' to 3', so that's kind of, you know, kind of a hint. And you will have to write the RNA sequence. You will need to also write the protein sequence based on whatever that RNA sequence is. I would say, think about this as an exercise. It will not be that straightforward because, as I said, I'm only going to give you part of a sequence. I'm not going to give you the entire sequence. So you might be given some of these nucleotides in a box, some other nucleotides in a box. You're not going to be given the orientation, so you will need to know the orientation. The way you will know the orientation is because you will have to complete the mRNA sequence to tell what the orientation is. And then given whatever, once you fill in the RNA sequence, again, I'll just give you a few. It's almost like, what is that game where there's this game where, you know, where they give you letters and then you have to fill it in. What is that called? Like, isn't there a game? Isn't there a TV game show that they give you a few letters and then you have to, not
Fortune?
Pardon?
Like Wheel of Fortune?
Wheel of Fortune, exactly. Yes, it's something like Wheel of Fortune, right? Where you're going to be given a few of these nucleotides and then you need to fill in the other strand and then you need to fill in the mRNA and then you need to fill in the protein, right? So that is what you're going to be given. I'm telling you, this is no surprise there, okay? But you'll be given this table, okay? So that's kind of what and that, again, then this brings, you know, all the way from DNA to mRNA to protein, all of this together, okay? The last lecture, which was lecture 16, this was really a lot of genomics and approaches. So they will get integrated to some extent into saying what technique you would use for this or what technique you would use for that. I think understanding what is an ORF, what is the features of an ORF, right? And there's two key things that make an ORF an ORF. Two approaches for genome sequencing. The whole details of this, do not focus on the whole details unless you plan to, unless you are graduating and you're getting a job at a sequencing company, then I would say, yes, you probably want to understand the steps of it. But the idea being what I would think you guys should focus on is the two ways, right? And one of them is really mapping and then sequencing, and then the other way is sequencing and then mapping, right? Large scale approaches to monitor gene expression. Know what those are. How do they compare to smaller scale methods, right? You'll need to know that. You'll need to compare and contrast, or at least list, you know, some of these methods. What are you measuring when you're doing a transcriptomic analysis, right? Are you measuring DNA? Are you measuring protein? Are you measuring a single gene? Are you measuring protein abundance? Or transcript abundance, right? So knowing what you're measuring is important. And also, if I say you want to study this single gene in the genome, what method would you use? You don't want to tell me that you're going to use a large scale method, because then I'm going to say, okay, how much is it going to cost, right? So do not tell me that. So if I say you have a single gene, and this is not, I'm telling you this because this is not something that happens with just undergrads, right? We also do this, like we have questions, I teach a graduate class as well. And every time when I ask, okay, what experiment are you going to do to answer this question? A lot of students like to tell me these very large scale methods. It's tedious, it's expensive. You don't even want to spend your time doing it. It doesn't mean it's wrong. It just means that it's not the best solution for you. So I will specify, if you had to look at the expression of a single gene and you have no money to do this research, what would you do, right? And then you want to think of a smaller scale method for these type of questions, these type of answers. Compare small scale methods to large scale methods for doing protein DNA interactions. So all of those small scale methods we talked about previously, filter binding assay, EMSA, DNA's footprinting assay. Those are small scale methods, even though DNA's footprinting is not really small scale. I mean, it's kind of considered medium scale maybe, but those are small scale methods. Larger scale methods is what we talked about just in this very last lecture, where we talked about doing ChIP-seq experiment or doing a chip, yeah? So that would be the method you would do to study protein DNA interactions, or if you want to see if your histone proteins are interacting with your DNA, yeah, you would do a chip experiment for that. Method to determine if your mRNA is being translated. There was just one slide on that, right? Looking at translation, because remember, you have all your mRNAs. We don't really know if those mRNAs are actually even going to be translated. We have no idea, right? So we have to also be able to think about, is your messenger RNA being translated? Because you might see you have your mRNA, you did your qPCR or whatever, and you can see your gene is expressed, but then when you try to look for the protein, you don't have any protein. You can't figure out. So something went wrong between that transcript, that expression you're seeing, to the protein. And it could be that it was either degraded, it could be the protein was made and it was degraded, or it could just be that something is wrong with translation. So what is happening at this translation level, and how can you detect translation? And we talked about ribosome profiling, so focus on that. Knowing the difference between, knowing how you can detect the presence and abundance of your protein, right? We talked about the Western blots and how that's what you're able to do. We didn't talk about large scale methods. I mentioned it towards the end, but there are large scale methods to look at protein abundance and the presence of a protein. But Western blots is what's normally used in all biochemistry labs or in any molecular biology lab that's working with protein. It's just doing Western blots. And these three are very similar, Southern, Northern, and Western. So thinking about how, what are you measuring in each of these, right? What are you measuring when you do a Southern blot? What are you measuring when you do a Northern blot? And what are you measuring when you do a Western blot? So all of that is important. You can expect to have a question where we're pulling all of this together from lecture 1 through lecture 16, right? So you can expect to have a question where I say, okay, here's a gene. I really would like to identify what is the function of this gene and what else in the genome this gene is regulating, right? So I will specifically ask you these particular steps, and this is just an example, right? Like, what is the method to obtain the gene? How are you going to get the gene to even be able to study it? It's there in the genome somewhere, right? How do you get it? Yeah. And how would you confirm once you've cloned this gene? How are you going to confirm that you have the correct sequence of the gene? What are you going to use to do that? How are you going to check to see, okay, you have the gene, is my gene expressed? Because maybe there's a mutation, maybe there's something goes wrong, maybe this gene doesn't get expressed. How are you going to check if your gene is expressed? How are you going to check that the mRNAs and transcripts are being translated? Again, pulling in all of the techniques we've spoken about so far. And how are you going to check if you have a functional protein that's produced? Methods to identify if you were to mutate this gene, how would you know what other genes in the genome it's changing the expression of, right? So you want to think about a larger scale method, right? So you're looking between your wild type and your mutant gene. So you want to see genome-wide, what are all the genes that's being regulated? And to think microarray RNA-seq type of experiments here with your mutant and your wild type, or your normal gene. And then methods to identify all the targets in the genome, right? So you say, okay, I know that it's changing the expression of all of these genes. What are some of the direct targets as well in the genome, right? Because often these two might not correlate, and I don't want to get into detail of why that is, but quite often the genes that are altered in expression level are not directly bound or interacting with your particular protein. There's lots of indirect regulation that happens in the cell. So don't worry too much about that. So what method would you use to think about which targets in the genome are being regulated? So this is kind of all pulling it together. These are the type of questions you would see in those 6 to 8 large scale questions in part C that you would need to be able to answer. But I think that's it. That's the last slide of things that I want to talk about that I want you guys to focus on. Any questions?
Professor,
Yes.
sorry. Could you just for the ability to start the section, can we just start any section?
It's gonna be exactly like exam 2, so all of them will be available all at the same time.
Perfect.
Yeah.
All right.
And you also will sorry, let me stop. Thank you for reminding me